morning, everyone. It's a pleasure to be here today to present some of the genome-based reverse vaccinology approaches that we've been using to identify novel vaccine antigens. But firstly, if we look back to the vaccines that were licensed during the 20th century, the majority of these were developed using Pasteur's principles, which are essentially to isolate, inactivate, and inject the organism causing disease. And as a result, we have many killed vaccines, live attenuated or subunit vaccines, all of which have had an enormous impact on the incidence of infectious diseases. And for example, it's estimated that there's been a 97 to 100% decrease in the incidence of diseases such as smallpox, polio, measles, and tetanus, just to name a few. However, despite these success stories, there remain several limitations to conventional approaches for vaccine discovery, and this is particularly true for organisms that are difficult to isolate or culture, or for those that are highly variable. And given that traditional approaches typically focus on characterization of only a few antigens at a time, as a result, this process is time-consuming and often it has proven to be unsuccessful. However, with more recent and continuing technology revolutions, the vaccine field has dramatically changed. And in particular, the ability to sequence the entire genome of the pathogen led to the idea of using this genetic information to identify novel candidates that have been missed by conventional vaccines. So essentially, the ability to look at the genome sequence and using a process that we called reverse vaccinology, we're able to evaluate all the potential antigens encoded by a pathogen. And as a result, many new vaccines can be discovered for pathogens for which conventional approaches have proven insufficient. So now, reverse vaccinology was first applied to serogroup B, Neisseria meningitidis, and just to give you a brief overview of meningococcal disease, there's over 1 million cases per year, and it's estimated that there's approximately 135,000 deaths. And the majority of these cases are caused by 5 main serogroups. And meningococcal disease is characterized by rapid onset and progression to sepsis or meningitis. And unfortunately, despite the use of antibiotics, there remains a high level of morbidity and mortality. So due to all these reasons, vaccination is considered essential in order to control meningococcal disease. So currently, 4 of the 5 main serogroups, A, C, W, and Y, can be prevented using vaccines that consist of the capsule polysaccharide that's conjugated to a protein carrier. And these vaccines are efficient in protecting against disease in all age groups. However, this approach hasn't been able to be used for serogroup B because the capsule polysaccharide shares homology to structures that are found in the host and as such is poorly immunogenic. And consequently, the majority of vaccine efforts have focused on the outer membrane structures of the meningococcus that have been isolated during the last 3 to 4 decades. However, most of these surface structures are highly variable and antigenically diverse or poorly immunogenic. So in light of all these obstacles, we decided to use a novel approach to target serogroup B meningococcus, exploiting the genome sequence of the first meningococcal strain and the first MenB strain. The genome sequence was completed in 2000. And essentially, starting from the genome sequence, which contains over 2,000 genes, we use bioinformatic tools to identify approximately 600 open reading frames that potentially encode novel surface-exposed or exported proteins and as such are potential targets for the immune system. Following this, 350 proteins were successfully expressed and purified from E. coli, and these were used to immunize mice. And the mouse sera was then used to confirm the surface localization of the proteins by flow cytometry. And more importantly, the sera was tested for its bactericidal activity. Now, from this selection, from this screening process, the top candidates were then further analyzed for their conservation between different strains. And also the antigens were optimized for their immunogenicity by creating fusion proteins that were then tested individually or in combination and in many different formulations. So essentially, the best combination that we chose was that which gave the broadest cross-bactericidal protection against diverse strains. And this composition has been called the 4-component MenB vaccine. It contains 3 recombinant proteins, which are 2 fusion proteins and a single polypeptide that's been combined with an outer membrane vesicle component. And this outer membrane vesicle component was the main antigen in the MenB vaccine that was successful in limiting the prolonged epidemic of serogroup B in New Zealand during the last decade. And so all these 4 components are combined in a suspension. And in addition to their immunogenicity and ability to induce bactericidal antibodies, all of these antigens have subsequently been identified as being important for meningococcal virulence or survival. So firstly, NAD-A is a nicerial adhesin, and it's involved in adherence to and invasion of host epithelial cells. FHBP is a factor H binding protein, and binding of factor H, which is a complement protein, enhances the ability of the bacteria to survive in the blood. NHBA is a nicerial heparin binding antigen, and once again, binding of heparin by this antigen may increase the ability of the bacteria to survive in serum and blood. And then finally, POR-A, which is the major component of the outer membrane vesicles. It's a porin which is involved in solute transport across the membrane. So now just to summarize this first section on MenB reverse vaccinology. MenB is an elusive target that's genetically diverse and variable. However, the 4-component MenB vaccine was created using the innovative approach of reverse vaccinology, starting from the genome. And this vaccine includes 4 components that are important for survival and or virulence of the meningococcus. And clinical trials using this vaccine in infants and adolescents have shown it to be immunogenic and have an acceptable tolerability profile. And essentially, to sum it all up, 11 years after the publication of the genome, a license application was submitted to the European Medicines Agency. And Peter Dull will present the clinical data of this vaccine in the next session. So given the success that was obtained with the MenB project, reverse vaccinology is now a routine approach that's used for vaccine antigen discovery. And furthermore, it's evolved to include multi-genome analysis to address the issue of species diversity and variability that remains a hurdle for vaccine development. And I'd like to give an overview of the approaches used for 2 additional pathogens, so the pneumococcus and pathogenic E. coli. So firstly, Streptococcus pneumoniae is a major cause of morbidity and mortality worldwide due to diseases ranging from otitis media, pneumonia, meningitis, and septicemia. And while there are several vaccines available, none of these afford universal protection. The 23-valent polysaccharide vaccine is ineffective in children under 2, and the multivalent conjugate vaccines still only cover a limited number of strains and serotypes. Therefore, there remains a need for a protein-based vaccine that could protect against globally circulating strains regardless of their serotype. So to this end, we used a combination of different genomic-based techniques to identify novel candidates. Once again, reverse vaccinology was used to identify putative surface proteins, surface-exposed antigens from the genome sequence. This was also complemented by using library immunoscreening to identify antigens that were recognized by convalescent sera. So this lets you identify proteins that are both expressed and immunogenic during infection of the host. And the sera used was both from patients recovering from pneumonia or meningitis. And then finally, the gene variability was assessed by using genome hybridization analysis to identify conserved antigens. So the combined use of these 3 approaches enabled the list of candidates to be refined and optimized before going into in vivo and in vitro screening for protection. So essentially 80 candidates were chosen from these approaches, and sera to these candidates was then tested using vaccinalysis to confirm the surface localization of the proteins. The sera was tested for its ability to kill the bacteria in opsonophagocytosis assays, and the candidates were then tested for their ability to induce protection in different mouse models of infection. And here you can see a summary of all these screening approaches, and several antigens were identified that were well conserved between the different serotypes, that were surface exposed, and that gave protection against at least one of the different strains from at least one of the different serotypes. However, further investigation and evaluation of these antigens revealed that the best approach to achieve a broad protection against lots of serotypes was a combination of several of these antigens. And in addition to the antigens in the table that I just showed, the genome-based approach has also led to the identification of several novel virulence factors, including the identification of the pilus for the first time in Streptococcus pneumoniae. And the pilus is present in approximately 30% of strains, or 50% of the multiple drug-resistant strains. And this pilus consists of 3 different subunits, each of which have been shown to be highly protective. So once again, genome-based approaches have led to the identification of not only novel antigens, but also virulence factors that have helped understand the pathogenesis of the organism. So now moving on to E. coli. Pathogenic E. coli are responsible for over 2 million deaths per year worldwide and have annual direct costs in excess of $2 billion US. Now, unfortunately, pathogenic E. coli are extremely complicated in terms of the classification and pathogenesis. And as you can see, there are numerous different pathotypes described for the intestinal and the extraintestinal infections, and these cause diseases ranging from diarrhea and hemolytic uremic syndrome to urinary tract infections, sepsis, and neonatal meningitis. And this complexity has hampered vaccine development, and while several attempts have been made focusing at individual pathotypes, so far these have proven to be unsuccessful in developing an E. coli vaccine. Now, the complexity of E. coli and also the need for a vaccine was further highlighted by the antibiotic-resistant E. coli outbreak that was seen in Germany last year, where over 4,000 cases were attributed to an enteroaggregative E. coli that had gained several features, several virulence factors typical of an enterohemorrhagic E. coli. So to identify novel antigens for E. coli, we used a multi-genome subtractive reverse vaccinology approach, where we selected candidates that were predicted to be surface exposed from 3 separate extraintestinal genomes. And this list was further refined by choosing antigens that were common to all the available extraintestinal or XPEC genomes at the time. However, also was further refined by choosing antigens that were absent or present with low homology from the non-pathogenic genome strains that were available. And an additional criteria for selection was the identification of proteins from a surface proteomic analysis that focused on E. coli outer membrane vesicles to identify surface exposed proteins. And altogether, we came to a list of 229 potential candidates. And subsequently, several antigens were identified that were protective in a mouse model of sepsis. And if we look at the top candidate here, which is a putative lipoprotein, and looking at its protection in the mouse model of sepsis, in the top panel, we show survival, in the bottom panel, bacteremia, and after both active immunization with the protein or passive immunization with sera-arrays to the antigen, you see 90 to 100% protection in the animals and almost complete prevention of bacteremia. So after this, we went on to look at the distribution of some of these protective antigens in additional extraintestinal strains, as well as several intestinal genomes that were becoming available. And essentially we saw that several of these antigens were well conserved between all the different pathotypes. And furthermore, that there's no clear genetic segregation between the intestinal and the extraintestinal E. coli. And possibly, rather than facing the daunting task of developing a vaccine for each of these separate pathotypes, it may be possible to consider E. coli as a single, albeit very variable pathogen for which a universal vaccine could be possible by using a combination of some of these common antigens. So now, just in the last few minutes, I'd like to touch on the use of structural vaccinology and the way in which genomics, reverse vaccinology, molecular epidemiology, structural biology and epitope mapping are all being used together to identify and optimize new vaccine antigens. So if we take the MenB antigen FHBP as an example, this antigen is present in 3 non-cross-protective variants, where variant 1 is the most common variant seen and is also present in the 4-component MenB vaccine. And we asked ourselves if we could increase the immunogenicity of this antigen. So firstly, the structure of FHBP variant 1 was solved, and then epitope mapping using monoclonal antibodies that were specific for each of the for each of the variants was performed. And it was found that amino acids contributing to immunogenicity of variant 1, shown in red, or variant 2 and 3, shown in blue, are located in non-overlapping regions. So following this, more than 50 different antigens or chimeras were designed in which different variant 2 or variant 3 epitopes were grafted onto the backbone of the variant 1 FHBP antigen. And further testing revealed that one chimera in particular was able to induce very high levels of bactericidal antibodies against all of the variant 1 and most of the variant 2 and 3 expressing strains. So this example shows you how structure-based design is a powerful approach that can be used to optimize novel antigens to give a broadly protective immune response. Now, to conclude, if we look at the way that new technologies and vaccine development have evolved over the last 30 years or so, we've gone from empirical wholesale approaches to the development of subunit and conjugate vaccines. And then with the emergence of the genome era, we've seen even more developments in the vaccine field. For example, 10 years ago, we started using genomes for vaccine development, applying reverse vaccinology to the MenB project. And today, we don't start a project unless we have the genome sequence for at least one, if not several strains of the species that we're targeting. And quite possibly in 10 years, we won't start a vaccine project without knowing the structure of the relevant antigens. So now the main question that remains is how can we continue to use powerful new technologies that are emerging to continually optimize both the discovery and the development of novel antigens. And these new techniques, as I touched on, include structural vaccinology, but also synthetic biology, RNA platforms, novel adjuvants, and systems biology. And really, we need to gain a better understanding of the human immune response. So essentially, if we look at the time needed for the discovery and development of a vaccine, a lot of the approaches that I've discussed today have helped decrease this first discovery stage compared to conventional vaccinology. However, with emerging technologies, we need to continue focusing on the time needed for development of these vaccines. And I think for the future, systems biology and innovative trial design will have a great part to play in this. And I think the next speaker in particular will will go into more detail on the innovative trial design. And so finally, I'd like to thank you all for your attention, and I'm happy to answer any questions that you may have.
Do we have questions?
Thank you very much for that talk. I wanted to get your comment on and particularly in light of some of our experiences. We've been humbled first by the HIV. And by the way, I'm Hari Pujar from Merck.
Sorry, I can't hear you very well.
You can't hear me? Okay. Is that better?
Yes, thank you.
I'm from Merck. My name is Hari Pujar. Coming from some background of being humbled by a couple of major efforts. One is the HIV STEP trial, and then the staph vaccine, which unfortunately didn't work, which used some of these approaches actually. So I'm just wondering what your comment is on the level of understanding of the immune system today to be compatible with the structural and the genomic approaches on the antigen side.
Yes, so if I understood what you're getting at, I think when we started using these genome-based approaches, it was very much just trying to look at and evaluate all the potential outer-membrane proteins. Now we're really focusing a lot more on also finding the antigens that are immunogenic, and in particular using techniques like the immunolibrary screening. For the structural vaccinology, also the use of different monoclonal antibodies or antibodies that have been shown to be protective is also greatly helping the way in which we can engineer these proteins. So I think in the future the use of immunology and the way better understanding of what constitutes a protective antigen, what is important, will play an even greater role, but that's something that really still needs a lot of attention.
Other questions?
So I understand MenB and strep pneumo, and that they're all bad bugs, but E. coli you really require essentially for good health. So, and I understand you're focusing on the pathogenic, but how are you ensuring there's not collateral damage to the good E. coli in the gut with your work?
So probably I didn't go into enough detail, but also the reverse vaccinology approach we use, we called a subtractive approach. So really trying to focus on antigens that were either absent or present with low homology in the non-pathogenic strains. So, and also some of the antigens are present in some of the fecal isolates that we've started looking at consequently. However, this is something that we'll need to assess as we go forward. E. coli is definitely a component of the commensal flora. However, it is a minor component. It's estimated about 1% of commensal floras, less than 1% E. coli, but it's definitely something that before we go on to development we'll need to address.
Other questions?
So most of the reverse vaccinology applications have been with bacterial vaccines. Can you comment on what are the challenges on applying this to viral vaccines, and comment on any advances made in that area?
Sorry, using reverse vaccinology for viral approaches?
For viral vaccines, infectious diseases from viruses.
So essentially one of the biggest problems with bacterial pathogens is that their genomes are enormous compared to viruses. So this approach is really very useful when you have possibly 2,000 genes with MenB. For viral vaccines, for viruses where you may have 10 or so antigens, it's not as important in terms of finding novel antigens. However, the use of genomics is really essential in understanding the variability and the diversity of the different antigens. So it's sort of a different approach depending on whether you're looking at the bacterial or viral pathogens, but definitely still incredibly important to have the genome of as many variants as possible.
Anyone else? We have time for maybe one more question. And if no one else has one, how can public and private sector institutions work together to foster the development of this approach? Are there incentives that are needed or that have been particularly useful?
Well, one of the benefits of this approach is now that we have so many genome sequences available, and as the techniques are constantly being refined. From the MenB project, we pretty much took all the possible outer membrane proteins that we could imagine. Now we've got a much better understanding of what constitutes a good antigen, so it can be greatly refined, which may make it easier for developing countries or public, developing countries to use this technology and to have access to the genomes. However, also there are several groups, and also Novartis Vaccines Institute for Global Health works very closely with Novartis Vaccines in order to use some of these techniques to apply for pathogens that are specific issues for developing countries.